before you get it.
Okay.
Okay.
Okay. So, all right. So if you're recording, I start with the first one again. So this is based on the all F2 mapping results. So we generated the mice in about 2005, 2006. This is BXD2 cross with some of the BXD2 F1, then we back cross with the, it's a F1 making F2, but the F1 could be generated by BXD2 cross with B6 or cross with DBA2. So that was the F2 result we did, and Lulu helped with the genotyping, and now they redo the analysis. So these are the gene, chromosome 4 gene, because all of a sudden chromosome 4, as if it's a hot zone, and that in the new BXD2 N2, not really N2, BXD2 cross B6, or BXD2 cross with the B6, and chromosome 4 has allowed us to narrow down the region to the distal region of chromosome 4. And that's why with the new mapping analysis, he actually found a few chromosomes, chromosome 4, chromosome 12, and chromosome 17, I think. He identified 3 interesting loci, but he focused on chromosome 4, potential chromosome 4 genes. And I believe he did something. Otherwise, chromosome 4 has many, many genes, but somehow maybe these are the ones exhibit polymorphism, and that meaningful polymorphism between B6 and B6 and DBA2. So he narrowed it down to finally about 60 something genes in chromosome 4. And some of them are not real genes, so not identified as a gene. That's why I don't have a gene name. And then at this distal end, these are microRNA, so they're not genes either. So with that, you cut down further another 15 also, not real gene. Then overall, maybe 40 something genes. And within the 40 something genes, then I look at their protein expression level. Is either in immune cells or non-immune cells. And so with that, you see more than half of them have very low expression in immune cells. So I label as very low. So where did I get that information? I got the information from this place called GeneCar. So for example, this MMS22L, that it says very low immune cells, but based on our own single cell data, this is very high in the BXD2 germinal center, dark zone. So this is where I found those information. You go GeneCard, this is MMS22L, and you keep going, then it tells you everything about this gene. Not everything, a lot of information. But then you could see the tissue expression. This is RNA-seq data. And unfortunately, RNA-seq doesn't tell you what kind of immune cell. You could see in, I think in lymph node, in thymus. Unfortunately, I don't see the spleen. So, oh, here is spleen. So there's some expression. But then this is the protein expression. And then you look at it, you don't see B cell or other major immune cells. I don't know why. Is it because not done or very low? But anyway, it's very low. So if it's very low, I just label very low. So that's the way I just went to each gene and then When it's very low, I just label very low. And it's when it's very low that I have less interest in that kind of gene. But if it's high, then these are potentially, it will be directly impact the immune system. So I just put high in T cell and B cell based on that information there. Okay, so that's one information I get. And the next information I have is whether, based on our own observation from our single cell RNA sequencing data between BXD2 B cell versus the BX2 interferon receptor knockout. And you go through the list again, and again, many of them are not detected. Doesn't mean it's not expressed, it's just we did not detect. However, if we can detect and show something meaningful, that give us another level of confidence that these could be potentially the candidate. And to some extent, I think it's when it's very low, it's also not detected. So to some extent, I think it's quite consistent between what's in GeneCard versus what we found in the single cell data. So these are all not very low and it's not detected. When it says high in T and B cell, we found it high in T and B cell too. So only one or two that is not consistent. Otherwise, most of them, I think this one is not consistent. But otherwise, when they show high in B cell in D, it is in our single cell data. So I think that gives us further confidence.
Can you show an example of the single cell?
Yes, I will show. So I'm going to show you. For example, okay, so let's come to this one. I say this is actually a very good one, the Miss M1. So, okay, so let's go see Miss M1. I say high in IgD negative B cell, and in IgD positive B cell interferon receptor knockout is higher than the wild type. So let's look at our single cell data. So where's Miss M1? So let me, oh, here. Okay, so Miss M1. All right, so you can see this is transitional and this is knockout. This is wild type. So knockout does have more than the wild type. And this is the plasma. So wait a minute. Let me make sure. What am I looking at? The knockout. So I need to correct myself if I made a mistake. So this is wild type. This is knockout. And that's the, that one. Let me confirm. And where's the, okay, I think I closed it. Let me open it. Open file. Let me open it from another resource. It's wild type. This is knockout. And let's go to the gene. Okay, so that's MS MYS M1. Okay. This one to be short. Yeah, right. So this is knockout. This is wild type. So I think I did label it right. So I say high in IgD negative, but in IgD positive, knockout is higher than wild type. I think that's right. Okay, so with that, so it is a germinal center, and it's the other file. Oh boy, let me open it. So, oh, wait a minute, that's the same file, isn't it? Pre-GC area. but I think here is even more obvious that this is quite high here, and this is lower, right?
Right.
Right. So okay. So let's see what this gene is for. I think this one is very important. This gene's expression is stimulated by DNA and acts as a repressor for STING to suppress type 1 interferon. Suppress type 1 interferon production and action. Actually, I didn't finish the sentence. And so here's a paper about that, which I think is very important. So that means type 1 interferon actually suppress this gene as well. By suppressing this gene, it enables even higher induction of type 1 interferon response. So we don't know if this is the candidate, but at least this is one important mechanism that we saw. That here's a response. It does say this lower expression of this gene in the wild type enable a higher interferon response here. So yeah, so I think we should pay attention to this gene. That's overall, I think. And of course, this gene, there's right. So this is the paper about that. And of course, you have, yeah. So if you have a lower expression of this, you will have increase in autoimmunity. And this is fairly new from last year. So this is a new concept. Okay. So I think this is a good one. And then I especially like this one overall. And I think this one is interesting too. This is, if you don't mention it, you forgot. But now it does ring a bell. This is FAF1. This is the fast downstream signal gene. It enabled the apoptosis. So this gene high in germinal center dark zone. So it's a, you go to FAF1. And you see it right there.
Yes, dark zone.
Right, it's very unique for germinal center. And I said overall, some of the genes so germinal center dark zone specific that we did not know. For example, this ITGB3, this one is germinal center dark zone specific as well. You see?
Yeah.
You don't even feel I change anything, but it does say that quite a lot of genes here are so co-regulated in a way, even. In a way, it's a germinal center dark zone hot area in the chromosome.
That is interesting, right.
Right. So, although I don't see big difference here, but it does suggest it's all possible because it is so specific for germinal center. And there's another one. So both are very germinal. So I see a label, quite a few, and I don't see a big difference. That's why I don't make a big deal. But this one, you do see, at least functionally, this thing, this is one mechanism why you knock out the interferon receptor. That will have such a good effect because this gene is that you knock out the interferon signal. Interferon itself provides a positive feedback loop, but if you have this gene, it will stop that positive feedback. And that suggests
So not
that.
detecting means just you pasted the other ones in and nothing. They didn't show up.
Yeah, so let's say, okay, so this one, okay, let's say DAP1. I paste in all the other gene. The one I said not detected is not detected. DAP1. So DAP1 is not there. And I tried the other sample, it's not there either. So it's not just I tried both sets. So not there is not there. Yeah, and that's why it took so long, but at least I completed the table and narrowed down to the one. And
it's almost like you
right,
get candidate genes,
right,
just, you know, with the program or something, and plug them all in and search for them and see if they're detected at these.
right. So it does make it now, it's more and more possible that we might really find the one. And I
Right.
kind of like this one too, this WASP2.
Yeah.
This
The
is
single cell
mm,
is untargeted. It's like a microarray, but you can even see where it is.
right, that's right.
You know, you
That's
can see
right.
its potential of.
Right. So here's another one that's higher in the knockout that we would not have known previously.
Right.
And here in the, I don't know.
Seems like the combo of transcriptomics, that is single cell omics, and genetics, genomics, and
right. That's right. That's right. Make it now sort of.
we were going to do that. Remember
right.
the advanced generation, the 150 versus 50.
Right. That's right. So I think with all these, I would say that should be a goal. We should do it instead of not say not doing. We should do it.
Right, because no, I'm just thinking just in crosses too, but just compare the 50 to 150 generation.
Right.
That'd be kind of easy.
Right. That's right. The polymorphic gene, and then just transfer that to the BXD2 and see what happened. Then instead of breeding a new mouse, you just do that kind of approach with Joe and Chen-Ren there. This is, once we get to that step, I think we finished the paper. So